Good morning students, good morning everyone. My name is Divya Banu. Today I am going to explain about glycolysis. Glycolysis. Glycolysis is the first step of respiration. It is the first step of respiration. Respiration is the process where complex carbohydrates are break down into simpler molecules and release energy to do some work. In respiration, there are 4 sequence of steps are present. That is, glycolysis is the first most step, an intermediate reaction. Glycolysis, intermediate reaction, Krebs cycle, and electron transport chain. Through these series of steps, we liberate energy in the form of ATP. ATP is the energy currency of a cell. This ATP is derived from complex molecular substances, and those complex molecular substances are break down into simpler molecular substances like glucose, galactose and fructose. From glucose, galactose and fructose undergoes series of respiratory reactions like glycolysis, intermediate reaction, Krebs cycle and electron transport chain. Finally we get ATP, and it is the energy currency of a cell. Whenever energy is needed, the bond of a ATP is break down to release energy whenever the body needs energy. So, from where we get glucose? We get glucose and galactose, fructose we get from the complex molecular structure starch. Starch is prepared by the plant through photosynthesis, and it is stored through photosynthesis. It is prepared by the plant and stored in the form of starch, and this starch is ingested by the human being or animal or something, and this undergoes series of respiratory substrates. It is first break down into simpler carbohydrates, simpler monosaccharides like glucose, galactose and fructose. These are all monosaccharides. These undergoes series of respiratory reactions like glycolysis, intermediate reaction, Krebs cycle and electron transport chain to get ATP. Now glucose is derived from the starch. It enters the blood from the gut. Digested from the gut, it enters into the blood. From the blood, it enters into the each and every cell of the organ. So now glucose molecule is entered the cell. This is glucose molecule. This is entered the cell through some protein channels. It enters the cell. Since glucose is very unstable molecule, it may move out of the cell. In order to prevent that glucose is move out of the cell, in order to prevent that reaction, it must be phosphorylized. It is phosphorylized by the ATP molecule. One phosphate group is attached to the glucose to form glucose 6-phosphate. This is the first step of glycolysis, that is phosphorylation of the glucose. Introduction to the glycolysis. Glycolysis is the first step of respiration, as we discussed above. It is the first step of respiration. Occurs in cytoplasm, whether it may be prokaryote or eukaryote. Prokaryote or eukaryote. It occurs in cytoplasm of all living organisms. The scheme of glycolysis was given by Gustav Embden, Otto Meyerhoff and J. Paranas. By the name of the scientist, it also called as Embden-Meyerhoff-Paranas pathway, the EMP pathway. Partial breakdown of the glucose to two pyruvic acid molecules is known as glycolysis. Note down the partial, not the complete, partial breakdown of the glucose. Partial breakdown of the glucose to form two molecules of pyruvic acid is known as glycolysis. This glycolysis process does not depend on availability of oxygen. It does not depend on availability of oxygen. Whether it may be in the presence of oxygen or in absence of oxygen, glycolysis takes place. In the presence of oxygen, it is called as aerobic glycolysis. Aerobic glycolysis. In the absence of oxygen, it is called anaerobic glycolysis. In the aerobic glycolysis, pyruvic acid is formed. In anaerobic glycolysis, lactic acid is formed. Okay. Glucose in the first step is glucose is converted to glucose 6-phosphate in the presence of enzyme hexokinase. Hexose means 6 carbon containing sugar. Hexo means 6 carbon containing sugar, and kinase is the enzyme which transfers phosphate group from high energy molecule to another molecule. That is, high energy molecule is ATP. Here high energy molecule is ATP. From the ATP, the phosphate group is attached to glucose molecule to form glucose 6-phosphate. Hence, the enzyme name is hexokinase, and the reaction is phosphorylation of glucose. This is the first step of glycolysis, and it is always forward reaction. It never be going to reversed. It is a irreversible reaction. Since energy is expenditure here, it is a irreversible reaction. Phosphorylation of glucose transfers phosphate group from ATP to glucose, forming glucose 6-phosphate. In the presence of the enzyme hexokinase, glucose reacts with ATP molecule to give glucose 6-phosphate and ADP. Next step is isomerization. Isomers means, what is meant by isomer? Same molecular formula but different structures, those are known as isomers. Glucose and fructose are isomers. They have similar molecular formula but different structures. Here glucose 6-phosphate is isomerizes to fructose 6-phosphate. This reaction takes place by the enzyme phosphoglucose isomerase. This is also called as phosphoglucomutase in some texts. Next reaction is second phosphorylation. This is the third step of glycolysis. Fructose 6-phosphate, by the addition of another ATP, it is going to converted into fructose 1,6-bisphosphate and ADP will be released. Here, fructose 1,6-bisphosphate is a 6-carbon compound. Fructose 6-phosphate is also a 6-carbon compound. Fructose 1,6-bisphosphate is also a 6-carbon compound. There are 6 carbons in this compound: 1, 2, 3, 4, 5 and 6. In first and sixth position, phosphate group is attached. Since 2 phosphates are attached on this fructose, it is known as fructose 1,6-bisphosphate. Here another ATP is utilized. For another phosphate group is attached to fructose 6-phosphate to give fructose 1,6-bisphosphate. Here phosphate group is attached to 2 different carbon atoms, hence it is known as bisphosphate. It is not a diphosphate. It undergoes in the presence of enzyme phosphofructokinase. Here again kinase enzyme. Kinase is the enzyme which transfers phosphate group from high energy molecule to another molecule. Next step is cleavage. Here fructose 1,6-bisphosphate is cleaved to form dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate DHAP and G3P. Fructose 1,6-bisphosphate is a 6-carbon compound. DHAP is a 3-carbon compound and G3P is also a 3-carbon compound. Here fructose 1,6-bisphosphate 6-carbon compound is breakdown to 2 molecules of 3-carbon compounds, that is DHAP and G3P, dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. It occurs in the presence of the enzyme aldolase. Next step is isomerization of the DHAP. In order to continue the glycolysis step, DHAP must be converted into G3P. DHAP is isomerized to G3P. It occurs in the presence of the enzyme triose phosphate isomerase. Since 3 carbon compounds are involved here, hence it is named as triose phosphate isomerase. Dihydroxyacetone phosphate is isomerized to glyceraldehyde 3-phosphate. Now 2 G3P molecules are available for every single glucose molecule. For every single glucose molecule, 2 molecules of G3P are involved here. Next step is oxidation and phosphate addition. In the next step, glyceraldehyde 3-phosphate is going to G3P is going to lose 1 hydrogen atom and it gives to the NAD. nicotinamide adenine dinucleotide. Here, one hydrogen atom is transferred to NAD to form NADH. And one phosphate group is attached to glyceraldehyde 3-phosphate to form, and another phosphate group is attached to glyceraldehyde 3-phosphate to form 1,3-bisphosphoglycerate. Here, for every one molecule of glucose, 2 NADH are formed. 2 NADH are formed. It undergoes in the presence of the enzyme glyceraldehyde 3-phosphate dehydrogenase. Here, hydrogen group is removed and it gets oxidized, hence it is known as the hydrogenase enzyme, dehydrogenase enzyme. Glyceraldehyde 3-phosphate dehydrogenase. Since here oxidation is taking place, removal of hydrogen from the compound is known as oxidation, and phosphate group is attached to this, hence the name of the reaction is oxidation and phosphate addition. 1,3-bisphosphoglycerate is formed. First ATP formation, substrate level phosphorylation. Substrate level phosphorylation. Here, transfer of phosphate from 1,3-bisphosphoglycerate to ADP. Transfer of phosphate group. Here 2 phosphate groups are present. Transfer of one phosphate group from 1,3-bisphosphoglycerate to ADP, forming ATP and 3-phosphoglycerate. Here first carbon position phosphate is transferred to ADP to form ATP, energy rich molecule. Hence it is known as substrate level phosphorylation. It undergoes in the presence of the enzyme phosphoglycerate kinase. Here and again the enzyme kinase, transfer of phosphate group from high energy molecule to another molecule to form ATP. 1,3-bisphosphoglycerate reacts with ADP to form 3-phosphoglycerate and ATP is produced. Relocation of the phosphate. Here 3-phosphoglycerate is present, 3 carbon compound, 3-phosphoglycerate. 3 carbon compound, 1, 2 and 3 carbon compound, 3-phosphoglycerate is present. At third position, 1, 2, 3, at third position phosphate group is present. This phosphate group is relocated to second position of the second carbon position of the glyceraldehyde 3-phosphoglycerate and form 2-phosphoglycerate. At third position phosphate should be present, but it is relocated to second position to form 2-phosphoglycerate. Moves the phosphate group from third carbon to the second carbon, converting 3-phosphoglycerate to 2-phosphoglycerate. It occurs in the presence of the enzyme phosphoglyceromutase. Dehydration. Removal of water molecule removes water molecule from 2-phosphoglycerate, resulting in phosphoenol pyruvate. Removes water molecule from 2-phosphoglycerate. Removes water molecule from 2-phosphoglycerate, resulting in formation of phosphoenol pyruvate. Removal of water molecule is known as dehydration. It occurs in the presence of the enzyme enolase. And last reaction is second ATP formation. It is also a substrate level phosphorylation. Here transfers the phosphate from phosphoenol pyruvate to ADP, generating ATP and pyruvate. From PEP, phosphoenol pyruvate, phosphate group is here from PEP, phosphate group is attached to the ADP to form pyruvic acid. And from the pyruvic acid, the respiration step, second step of the respiration continues. Okay, before going to the bits, we'll once again revise these steps. Glucose is converted to glucose 6-phosphate, and from glucose 6-phosphate, fructose 6-phosphate is formed. Glucose is converted to glucose 6-phosphate in the presence of the enzyme hexokinase. 1 ATP is utilized here. From glucose 6-phosphate, glucose fructose 6-phosphate is formed. First reaction is irreversible step, second step is reversible step. From fructose 6-phosphate, by another addition of another molecule of ATP, fructose 1,6-bisphosphate is formed. It occurs in the presence of the enzyme phosphofructokinase. From fructose 1,6-bisphosphate, it undergoes cleavage reaction to form two molecules of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, G3P and DHAP. It is a 6-carbon compound, and G3P and DHAP are 3-carbon compounds. It occurs in the presence of the enzyme aldolase. Glyceraldehyde 3-phosphate, from glyceraldehyde 3-phosphate, DHAP is interconvertible to G3P. In order to continue the glycolysis reaction, DHAP must be converted to G3P. From each glucose, 2 G3P molecules are available now. Glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. It is a dehydrogenation reaction and addition of phosphate group. To glyceraldehyde 3-phosphate, one phosphate group is attached and hydrogen group is given to the NAD to form NADH. For each G3P molecule, each NADH is formed. Since 2 G3P are present, 2 NADH are formed. 1,3-bisphosphoglycerate is formed. From 1,3-bisphosphoglycerate, one phosphate group is given to ADP to form ATP. This is the substrate level phosphorylation. It occurs in the presence of the enzyme phosphoglycerokinases. 3-phosphoglycerate is formed. 3-phosphoglycerate is converted to 2-phosphoglycerate. Here, position of the phosphate group is changed. Third phosphate group is present at third position. It is changed to second carbon position. It occurs in the presence of the enzyme phosphoglyceromutase. And the reaction from the 2-phosphoglycerate, water molecule is removed from the 2-phosphoglycerate. It is a dehydration reaction. Since water is removed, it is known as dehydration reaction. Phosphoenol pyruvate is formed. PEP is formed. From PEP, another substrate level phosphorylation occurs in the presence of pyruvate kinase. Another molecule of ATP is formed. Here ATP is formed. From phosphoenol pyruvate, one phosphate group is given to the ADP to form ATP, and 2 molecules of pyruvic acid or pyruvate is formed. Here product of glycolysis is 2 molecules of ATP, 2 molecules of NADH and 2 molecules of pyruvate. Here when we see these reactions, 4 molecules of ATP are produced. Since it is an energy expenditure step, since this is the energy expenditure step, 2 ATP molecules are utilized here and 4 ATP are formed here. 4 minus 2, net ATP is 2 ATP. Although 4 ATP are formed, 2 ATP are consumed in the above reaction. Only 2 ATP are net gain of the glucose and 2 molecules of NADH and 2 molecules of pyruvate are formed. Now we are going to discuss the bits. First bit is glycolysis occurs in. As we discussed, we are going to see the options. Options A cytoplasm, B mitochondria, C peroxisomes, and D chloroplast. Option A is correct. Glycolysis occurs in cytoplasm. Which of the following statement regarding the process of glycolysis is correct? Option A, glucose undergoes complete oxidation to form 2 molecules of pyruvic acid. By undergoing elimination reaction, we can easily get the answer. And since we discussed that glucose is going to undergo partial oxidation, not a complete oxidation. It is a partial oxidation of glucose to form 2 molecules of pyruvic acid. Here, so option A, option C are false. Complete reaction. It is not a complete oxidation. It is a partial oxidation. From B and C, since we know that 2 molecules of pyruvic acid are formed, so partial oxidation and 2 molecules of pyruvic acid are formed. Not a 1 molecule, 2 molecules of pyruvic acid are formed. Then option D is correct. Next question, third. In which of the following reaction of glycolysis a molecule of water is removed from the substrate? Fructose 6-phosphate to fructose 1,6-bisphosphate. Option A, fructose 6-phosphate to fructose 1,6-bisphosphate. It is not true. 3-phosphoglycerate to 1,3-bisphosphoglycerate. It is not true. Phosphoenol pyruvate to pyruvic acid. It is the last step of the reaction. Dehydration reaction is not going to happen here. We know that last but one step, one molecule of water is removed from 2-phosphoglycerate to form phosphoenol pyruvate. This is the dehydration reaction. Option D is correct. Glucose is converted to glucose 6-phosphate due to the enzyme. Glucose is converted to glucose 6-phosphate due to the enzyme. Option A, hexokinase, B, invertase, C, hydrolase, D, phosphatase. Phosphatase is involved with the phosphorus group. Hydrolase is involved with the removal of or adding of water molecule. Invertase is inversion of the compound. Hexokinase is the right answer. As we discussed in this lecture, glucose is converted to glucose 6-phosphate in the presence of the enzyme hexokinase. Kinase enzyme is involved in transfer of phosphate group from high energy molecule to another molecule. Since 6-carbon sugar, hexose. So hexokinase is the right answer. I hope you like this video. Thank you for watching this video. Thank you.